What if you could see beyond the surface, right down to the secret choreography happening in every molecule? Today, we sit down with Dr. Brad Swarbrick, global leader in near and mid-infrared spectroscopy, whose work shapes how the pharmaceutical world measures, understands, and controls its most complex reactions. With over 25 years of experience, Brad has helped some of the world's largest pharmaceutical and medical companies map out new control strategies using PAT methodologies. He's not just a scientist, he's a collaborator, a mentor, and the editor guiding the conversation at the Journal of Near-Infrared Spectroscopy. Whether it's developing robust data analysis tools at KAX Group or mentoring the next generation of chemometrics, his approach is quietly transformative. Thanks, Brad, for joining us for today's podcast.
Thanks, Lyndon. Thanks for the nice introduction.
Yeah, my pleasure. And it's really great to be able to speak to you today and to also learn the insights you have and the experiences you have gained over the years in PAT, chemometrics, and also in starting your own company, KAX Group. Maybe if you don't mind sharing a bit about your whole experience in chemometrics.
So I started my chemometrics journey back in 1993, doing an honors project with a new spectrometer called a Fourier transform infrared. They told us that this spectrometer could capture a spectrum in 30 to 60 seconds, and everybody's saying, no way, because we were used to mid-infrared taking 30 minutes to collect the spectrum. But the Fourier transform system opened up my eyes to a lot of potential. But the greatest drawbacks we had back in those days was computing power. We had 33 megahertz or 66 megahertz computer. So what takes me 30 seconds today took 8 hours back in those days to generate one PLS model. Yeah. And then you'd have to JCamp the data and put into Excel. And if it was wrong, then you'd take another 8 hours to generate another model. I started off there, and then after finishing my degree, I moved into a near-infrared spectroscopy company working in agriculture. Chemometrics was the glue that held it all together. And because I had that experience from my honors project, it was a natural fit to the companies. We were making spectrometers for measuring agricultural products, for developing online systems for application on farm, where during harvesting, the NIR spectrometer would be in the combine harvester. We can measure protein and moisture in real time. And that caught the attention of the pharmaceutical industry because they were moving into something called process analytical technology. And I was lucky enough to get a job with the early Pfizer PAT group based in Sydney. Again, very large spectrometers. They weren't really designed for process. So we had a massive learning curve. But over the years, the instrumentation started to get faster. Computing power started to get faster. Instruments were becoming smaller. They could generate a spectrum in 10 milliseconds rather than, say, 60 seconds with the old spectrometers that we were using. The adoption was, because it was unknown, adoption was really slow. I can still say adoption is still slow today. It wasn't going as fast as what we wanted it to. But after I finished with Pfizer, I moved to another company called Sigma Pharmaceuticals, developed their entire PAT program, did some work with fluid bed drying. I thought in 2008, maybe it's time just to go out there and pitch to the world what he can do. And I thought the Southeast Asian region would have been a good place to start. I worked for a pharmaceutical consultancy company trying to promote PAT. That didn't go as I expected it to, because again, there was a slow adoption. And that's when I moved into focusing more on chemometrics. And so I started a business in Australia called Quality by Design Consultancy. And I agented for one of the world's largest chemometric software providers. That led to them asking me to move to Norway, where I stayed for 3 years, helping to develop their business. Then I moved to the United States in 2013 and helped the company over there promoting chemometrics, developing different softwares. Then for family reasons, I moved back to Australia in 2015. And that's when I had the idea of starting KAX Group for looking at hyperspectral imaging. During the development of that software, we realized that we'd written the easiest to use Chemometrics software package in the world. So now we've got KXG as one of the top providers of Chemometrics software for pharmaceutical applications, but for an application. So there's always been an interest in Chemometrics from the start.
Nice. And yeah, I think like your whole experience and your whole journey really reflected a lot of how much the world has progressed. And also it's quite amazing that you've been able to catch on to all these different technologies that are happening and actually making use of them and applying them in the real industrial applications. Could you share a bit on this aspect when you were, I guess, initially even trying to promote PAT or get PAT? You mentioned that it was challenging, the adoption rate is slow. What were the challenges then, and are the challenges still present now?
I can say in the early days, when I was working for Pfizer, they had a dedicated PAT group.
Yeah.
It was one of the very rare companies that had dedicated PAT people. Our greatest challenges then were infrastructure. IT infrastructures weren't what they're like today. Data transfers were slow. Implementation of equipment. So we're talking about big grating-based mechanical equipment. Implementing that into process, you'd have to have refrigerated cabinets, vibration-proof containers, fiber optic probes that might have extended for 20 meters, things like that. And there was a lot of practical issues that prevented us from progressing. But with computing power just exponentiating in the mid, like in the late 2000s, the reliance on databases and greater speed of instrumentation, that's allowed us to do a lot more to bring the process, the spectrometer closer to the process, like equipment like this. Which
thanks.
Bringing something like this, you could have this sitting beside the process. Yeah. Compared to having a control room that's intrinsically safe barriers to the process, etc. Yeah. I mean, you've got the intrinsically safe barriers here as well. That was a much bigger challenge back in the earlier days. And PAT, the promise, there was an unrealistic expectation in PAT. The biggest, in my experience, people think it's bringing the laboratory to the process. That's quality by testing, not quality by design.
I see.
About timely measurements of the state of the process to ensure that the whole thing is running as it should be. And if something happens, have you got a window of opportunity to correct it without a deviation occurring or predict the deviation? A side benefit of PAT is quantitative analysis or quantitative outputs. Now they're good. If you can do predictions at the point of processing and you can validate them, well, you've eliminated a need for the QC laboratory to perform that particular test, and they could focus on more important tests rather than just trying to do routine analysis. There's always going to be a space for a QC laboratory. But what you're trying to do is, first and foremost, understand your process using PAT, and if you can get the side benefit of quantitative analysis, even better.
The challenges still present now? Or
the biggest challenge I've seen is that when a company wants to adopt it, they don't dedicate resource. They don't dedicated resource to it. They'll choose somebody with a bit of aptitude in a QC laboratory or a tech services laboratory, and they'll say, you're the new NIR person, but you still have to do your job. PAT is a dedicated resource. It's actually a team. So if you don't have that mindset, your PAT journey is just going to be a flat line compared to working with some of the bigger companies. Like one of the greatest projects I worked on was the Vertex continuous manufacturing system in Boston. That was a team of 20 plus people working 24/7.
Wow. Okay.
PAT was one of the key focuses. We were the software vendor for that, or part of the software vendors for that system. The success of their implementation was because they invested in people and in the right people to do the job.
Thanks. Yeah, I think that's really critical when trying to implement, whether it's new PAT or even new software architecture interfaces for chemometrics, or even just to control the overall aspect of it. And on that, when you were doing like software deployment, has the approach changed in production environments compared to the past and now? Are there more regulations, or are there more concerns on maybe even sustainability? Is that also something that needs to be also accounted for? Yeah.
So the IT structures have changed a lot.
Okay.
So that allowed PAT to go from being, in a lot of cases, at line
Mhm.
to now online or inline.
Okay.
And so you need a good, you need a good IT infrastructure, particularly talking to your SCADA systems, having your PAT management layer. And so a lot of companies were born out of that. They were making PAT management systems that sat just below the SCADA. And so in that way, you could demarcate quality from manufacturing in an electronic sense. Because in a pharmaceutical environment, the quality department and the manufacturing departments have to be separated. They can't. Quality can't make decisions on manufacturing. Manufacturers can't. And so the PAT management platforms allowed you to digitally demarcate that.
I see. Okay.
So data integrity has become the hottest topic in the industry over the last few years.
Yeah.
To the point now that the European Pharmacopoeia have just written a draft guidance on data management. So now the integrity, or who collected the data, we're generating a lot of data with smaller instruments. For instance, your instrument can capture 3 spectra per second.
Yep.
Over 2 or 3 hours.
Yep.
So yes, the infrastructures had to change, and the way we now analyze the data, having software interfaces that capture summaries of particular events, but also with limits on it, so that if we could see when the process was going to go out of control, we need a way of taking those outputs and speaking to the SCADA. And the SCADA says, okay, we're going to break this point. What do I need to do? It goes to its ladder logic and says, okay, I need to change this. Changes, and in real time, this feed-forward feedback cycle, which is something we couldn't have done years ago. Although the petrochemical industry had been doing it for many, many years.
Okay.
Pharmaceutical, there's been quite a few barriers which have slowly been broken down.
I see. Okay. And any of that related to more operational parts of it when dealing with the IT infrastructure? Is there something that always needs to be, I guess, discussed with and aligned with the IT department so that you can implement, for example, a software like that you offer, like,
IT is king.
Yeah.
In the pharmaceutical industry, the IT department is the king.
Okay.
A lot of that's handled by Windows, Microsoft Windows security. So you pass all of that security stuff over to your Windows authentication system. You just utilize the connection with that system. You provide a technical solution.
Okay.
That allows you to use the Windows authentication system. There's always been questions on in a process, in a manufacturing process, who owns the data? Well, the operator doesn't really own the data. And there's having roles and functions, so if they can log into the system, their credentials are logged to that data, but they don't actually own the data. They're following a set procedure that's been defined by a developer, and it's been approved by a QA department. We, as KAX Group, we've developed recently something called Vector Vault, which handles that. So in order for an operator to use our process interface, it's based on 3 levels: administrator, developer, and operator. And so an operator can only access methods that have been assigned to them by an administrator. So as soon as you start up the software, it makes sure that all the instruments are online and make sure that they're qualified. And when the operator pushes start on the process, it just does its thing. And so
that makes it a lot easier to manage.
Makes it a lot. Yeah, it's
the whole data.
It's taking advantage of Industry 4.0.
Speaking on Industry 4.0, what are like the new kinds of technologies or trends that you see that could be further used to improve on the whole like process industry? For example, Gen AI, do you see that being useful? Or
in process control applications, I see AI has a role to be able to make some sort of decision to change the process. But In terms of quality, and this is just my opinion, I think AI has no part in quality.
Okay.
It should never have any part in quality decisions. Its whole structure changes, and therefore trying to go back and work out what's happening, I see there's big problems with that.
Okay.
So for me, AI has great opportunities in changing the process as long as there's a validatable system on top of it that can verify that the change hasn't had an impact on quality. And that's where, again, you have the SCADA and you have the PAT management system. That should always be maintained on empirical models that can be validated, or mechanistic models that have got a theoretical basis to them.
So meaning if there are models that are taking advantage of the latest in machine learning, for example, but if they are stable, then it can still be validated if they're fixed. And then you have a way in your software of actually validating like models built by that approach, right?
So Chemometrics, like I've written textbooks with Professor Kim Espenson.
Okay.
Right? So he's probably the world's leading expert on sampling.
Okay.
And coming out of the Norwegian chemometrics background, validation is the most important part of any chemometrics model. And we always say test set validation is best. Test is best. Although you can do cross-validation, there's many different ways of doing. Those are only if you've got small data sets or you've just started. In order to ensure that the model is going to be robust in the future, we have to use independent test sets. And because a PLS model is a fixed, or a PCA model is a fixed entity, as soon as you say you apply it to the data, it will give you, if you push the predict button 10 times, it will give you the exact same result down to 10 decimal places. Yep. I've seen AI systems where you push the button 10 times and you get 10 different answers.
Yeah, that is a different, it should still be quantitative data based on like physical principles and theories behind it. Yeah.
The measurement is typically done at the highest point in the process. You've got your mixing boost going straight up into your system before it's even measured by the system. And then straight afterwards, you could put a sampling point. So you could capture the exact same sample as you've collected the spectrum from. You can't do that if you put a probe into the side of a vessel because you don't know what you're measuring. In cases like this, you can measure. And this is the sort of thing that we've been trying to promote to the industry for quite a while. We have to get that one-to-one sample to spectrum correlation, and the big, massive challenge. Yeah.
And besides this sampling being a challenge, is there also challenges that you've seen in spectroscopy, whether it's like or fouling? What are the common challenges that you see?
The old days, in the old days when you had grading-based instruments or older Fourier transforms, you'd have a lot of instability.
Okay.
A lot of those, like particularly with the grading-based instruments, you're taking a large light source, pushing it almost to its maximum capacity. Then you're trying to focus the light onto a 10 nanometer slit. And so you just lost 99.9% of your
I feel like
Then the Fourier transforms come along, and you took advantage of before the Jacquinot advantage, Pond's advantage, etc. But the thing is, they were slow, and Fourier transform instruments are notoriously noisy. That's why you got to do a lot of coalitions. So like the newer diode array and non-moving part spectrometers, you have a lot more robustness. So you don't have the full range of a research instrument. But, sometimes you don't need that. Resolution spoken about a lot. The resolution matters in some cases, but not in all cases. So I think miniaturization is the unknown sometimes.
Yeah, that's true. And I think that's something that always needs to be like overcome when a new, technology or something is being tried out in the market.